Good day and welcome everyone. I'm Jack Van Horn from the University of Virginia, and please allow me to welcome you to this edition of our 2020-2021 Foundations of Biomedical Data Science Seminar Series. This lecture is supported by UVA's Integrated Translational Health Institute of Virginia, the UVA Brain Institute, the College of Arts and Sciences, the School of Data Science, and through a grant from the National Institutes of General Medical Sciences. It is my pleasure today and privilege to welcome Dr. Daniel Gardner, Professor of Physiology and Biophysics from Weill Cornell Medical School in New York City. Dan Gardner has cultivated a very impressive career serving as the head of the Laboratory of Neuroinformatics and as Professor of Physiology and Biophysics at Weill Cornell for many years now. He has made enormous contributions to the synaptic biophysics underpinning networks of neurons in Aplysia, delivered resources for the exchange of neuroscientific data, and examined the parallels between biological and artificial neural networks. He and members of his laboratory like to ask the big questions, such as are neural circuits and the computations they perform evolutionarily conserved? The answers to such questions may be simplified as analyses of multineuron recording and inform new neuromorphic designs for future artificial intelligent systems. Dr. Gardner examines new approaches to explore this question, including a projected Neuromorphic Neural Networks, or Encubed collaborative, as well as something called the Next Cool Virtual Meeting, which I'm really interested to hear about, Dan. He has received numerous research accolades and research support from federal agencies such as the National Institutes of Health, making vital contributions under, for example, the United States Human Brain Project that took place during the Decade of the Brain, which is a kind of a precursor to the Brain Initiative today, as well as to the European Brain Project. Today's presentation from Dan is entitled, Do Biological Neural Networks Perform Identifiable and Verifiable Canonical Computations? As always, we are streaming this lecture live and for recording via YouTube. If you are watching via YouTube, thank you so much for joining us today. And also our specially selected 2020-2021 Biomedical Data Science Innovation Lab participants are encouraged to submit any questions for Dr. Gardner via the chat feature of their Zoom sessions. I will synthesize these questions and ask them on your behalf during the last 10 minutes or so of the lecture. And with that, welcome, Dan. Thank you so much. We are really excited to hear your lecture today.
And Professor Van Horn requested a football analog. So the UVA quarterback fades back, throws, and it's intercepted by the big red. Cornell now has the ball. All right, enough of the football analog. Now, I'm going to be asking this question, but I'm not going to be answering it. So I'm going to propose this neuromorphic neural networks viewpoint. And neuromorphic neural networks with the 3 N's gives us the logo that you can see on the right called Encubed. The ideas behind this come from a very, very large number of colleagues at many, many institutions over many, many years. Too many to thank individually, but I especially want to thank Chuck Stevens, who's been inspiring me since 1971. And among the many people I said who've thought about this question, including Chuck, well, we had a bunch of us about 25, 30 years ago now. And what we thought about it and what resulted from it is noted on some of the following slides, and it's expanded later. But first, we have a mandatory disclosure statement. My money comes only from federal grants. I'm a consultant occasionally to places in the US and the UK, but I do get significant income from these books. For instance, The Principles of Neuroscience, edited by Candelinar, I get 25 cents anytime one of you buys a copy. So I'm shamelessly going to be plugging this book throughout the talk. But the real self-serving motivation, not financial, is to encourage people in the data sciences, in computational neuroscience, and in the machine learning communities to think about the question of what computations are actually being performed by biological neural networks. It's an interesting question to which we don't have an immediate answer. I'm going to begin with a personal history of neural networks and what they've meant to me, which gives you a sense about why I've been thinking about this problem for so long. Artificial neural networks, for those of you who don't work in the field, were first proposed in ancient times in the 1940s, before people knew really anything about how neurons and synapses work. And they were model objects that had some of the properties that people thought neurons had. They were a little bit like logic gates that were on or off and had multiple inputs. And the field didn't go very well with the failure of what were called perceptrons, which it turned out couldn't solve some simple basic problems. But then in a classical past that was more recent than that, starting in the 1960s, these were made a little bit more neuromorphic, especially as layering became possible. And an artificial neural net didn't have just one or two layers, but multiple layers. They were very functional for simple problems, elegantly carried out, but they had a very, very non-neuromorphic backup, which a method for operation, which I'll talk about very briefly, called backpropagation as a learning rule. And as we will see, back in this classical past, a book that I and my friends put together said these might be more interesting if they were more neuromorphic. In the present, of course, artificial neural nets, due to the growth and processing power of modern computers and due to much more subtle and interesting design of the artificial neural nets, can perform some extremely important machine learning problems. And so they are enormously functional today, but they still cannot be one-to-one matched to what it is the brain does or how it does it. The future perfect that I list down here, it would be really nice to leverage our understanding of neurons and circuits to be able to build artificial neural nets that were more neuromorphic and so enhance machine learning. And it also might be useful to think about some of the very clever ways people are now designing artificial neural nets as we contemplate ways in which the brain does what it does. So going back, this is supposed to be historical. This is 1987. I took a ganglion I'd been working on since the 1960s, and I presented it on the right as if it were a neural network. But of course, it's biological. Instead of being arbitrary units emulated in software or built with transistors and capacitors, these were real identified neurons in a real ganglion of the marine snail Aplysia. And I presented this at the first International Conference on Neural Networks, just when the field was beginning to get interesting. And as you see, it was a multi-layer network with defined properties. Also around then, we had a meeting at Woods Hole in which this is the fanciful design for the t-shirt for the meeting, we thought we could link biological and artificial neural networks and look to see what their commonality was. And you can see the red biological neurons in the lower left and the artificial neural network units and connections in the upper right, coming out of something which is a meld between a hippocampus and an Aplysia, which were the subjects most often studied in this context. In 1989, and you see this was again early, this was the second volume of the journal Neural Networks, I proposed testing an artificial neural network learning rule in a biological neural network. It failed spectacularly, but it was still interesting enough to be published, thereby underscoring the idea that artificial and biological neural networks had some real fundamental differences. I was not the only one who began to point this out. From the field of neurobiology, you can see some names on the left. The one up at the top, Dr. Gordon Shepherd, published a paper in which he pointed out some very, very significant differences between biological and artificial neural networks. And other investigators, again in the 1980s, listed down below on the left, contributed to ideas for this birth of neuromorphic neural networks. Even Francis Crick got into the act, and he published this commentary in Nature at the end of the 1980s. And you can see in the center, the lower part of the center column, he points out that whatever it is artificial neural networks do, the brain is different in almost every respect. And that's Francis quite elegantly pointing out how far one has to go to be able to talk about, to linking the artificial and the biological neural network. So after this this flurry of interest by neurobiologists in artificial nets, and even interest in the people who were purely developing artificial nets in the brain, I got together some friends and we asked this question: is a network of neurons really a neural network? We noted back then that some features of artificial neural nets and the biological networks of neurons, those are the BNNs you've seen, resembled one another, but others didn't. Now, my co-authors then were leveraging ANNs to understand BNNs in the hope of making the artificial nets more neuromorphic and more capable. But in the intervening 27 years now, we know much more about biological nets. And the sophisticated artificial nets, deep convolutional nets, long short-term memory nets have become much more useful. So it's time to revisit this, and that's what I'm doing today. I'm going to ask some artificial neural network inspired general questions about how biological neural nets compute, and questions about whether evolution has conserved some of the basics for this. Back in 1993, my colleague Eberhard Fetz pointed out, in the context of a biological net, how artificial neural nets did things in terms of excitatory and inhibitory connection matrices, which had variable strengths, something that sounds very much like the way biological nets work. The problem that we stressed in 1993 was that artificial neural nets and their properties are summarized by the diagrams on this slide and the equations that accompany them. Biological networks work a little bit like artificial nets according to the scheme on the left. That is, they take in inputs, they sum it, there's a nonlinear transform. This is then transformed along an axon in a forward direction from cell body to termini, and then there are connections made on still other neurons. And everything on the left is sort of plausible in a biological context. But on the right, artificial nets require backpropagation of errors. And multi-layer backpropagation or local tuning of synapse strength by global error signals, or error signals many, many layers further up, these were viewed quite skeptically in 1993. As I'll show later on, the skepticism has been a little bit reduced, but the basic backpropagation scheme shown here on the right, which I'm not going to go into in detail, seemed very different from the way we thought neurons worked in 1993. Now, we should remember that evolution works. Evolution designs neural networks. Neural networks work, and I'm also going to stress the fact that neural networks process or compute information. Of course, they evolved to be able to learn, but what they are learning to do is to perform pattern recognition, to be able to recognize, say, a piece of fruit from a predator, to classify many, many different inputs, to make decisions on the back of it, and to form action patterns as a result of this. We know that the networks in our brain and even in other animals and even in other parts of our nervous system, like the spinal cord or the eye, may be optimized for specific features of the real world because evolution found that to be efficient. So things like locality, low dimensionality, relevance, novelty, those things may uh, get more attention from nervous systems than others. Some biological neural networks, and by this I mean small networks composed of a manageable number of neurons, some of them do things that we can understand. Integrators, for instance, we can understand. Comparators. Some use maximum entropy strategies for efficient coding, but in general, we don't know how they work. That is, we don't know what they're computing or how they're computing it, and that's a motivation for this work, because if we did know that, then perhaps the ways in which our brains efficiently do this computation and reduce error, making use of only the mechanisms that the laws of physics and evolution make available to them, could that enhance the design of artificial neural networks, especially artificial neural networks that we want to design to solve problems that are similar or isomorphic to the ones that our brains solve. Now, I use the word computation, and we probably don't think of our nervous systems as computing, and we may think of arithmetic, for instance, as something that's very, very artificial that we can do, but certainly our nervous systems didn't evolve to do. And so I'm just pointing out that many lower organisms, in addition to people, seem to be able to do computations. There are reports that pigeons can behaviorally show subtraction, understand subtraction, and modify their actions in a trained situation according to carrying out. Even something as simple as an insect, honeybees, can use tags for something that's greater than what they've seen before, or one fewer than what they've seen before. And they can again modify their actions uh, by this. So nervous systems, even what we used to call primitive nervous systems, seem to be able to actually do computation. And of course, I'm using the term in a much larger sense than that. I'm using it to mean processing information, but I find it striking that computation itself, counting, is something that nervous systems seem to be able to do. Now, here's a very famous slide that most of you are likely to have seen before. And this shows from the laboratory, from Dan Yeamans working in the laboratory of Jim DiCarlo at MIT some years ago. This shows similarities between the circuitry that does processing in the ventral stream of primate vision, and that's shown up on the top, and an artificial neural network trained to recognize people from photographs of those people. And what's striking about the similarities is that in each case, these are composed of simple units, neurons up above and artificial units down below. There is a feed-forward organization to these. There are nonlinear transforms, and although the two are very, very different, what is striking about this is that at several layers of both the real biological net and the artificial net, there are individual units and sets of units that seem to have the same selectivity for the very same pictures in the very same way, in different ways at different levels. And the maturity of being able to recognize an individual based on information that is processed across the multiple layers, that also progresses in both the biological net and the artificial net. So this reminds us that there are enough similarities between what a multi-layer system can do to make this an interesting and important question, even though the behavior at the individual levels is very, very different for the biological and for the artificial system. So let's ask ourselves, that's enough of an introduction. Let's ask ourselves, what is it that biological neural networks are computing? Well, I don't know. These are unanswered questions. These are very reductionist questions. These are the simplest way I can express it. And one question is, what is it that brains are computing? Then how, if you know that, how are those computations actually implemented by neural circuitry? And what to me is the most interesting potential question is the third one. Do these computations form a small canonical set? That is, if evolution has conserved as it has the basic properties of neurons, synapses, glia, across the animal kingdom, as well as the goals that nervous systems produce, is the next level, the circuit level, also conserved? And is there a small canonical set of circuits or computations performed by this circuit that one can identify comparatively by looking at different parts, say, of a single nervous system like the cortex, or by looking across nervous systems, even perhaps to some of those honeybees and Aplysia that I mentioned earlier? And then there's a fourth unanswered question, which is neurons are cells, glia are cells. They make use of neurochemicals. And which of the many, many properties that characterize these cells that happen to be neurons are actually the ones that are essential for computation? There's another question not listed here, which I may touch on briefly, which is, how do biological nets solve the curse of dimensionality? Because they have far more parameters to adjust than artificial neural nets do. And I think these really do qualify as some of those elusive principles of neural science that we would all like to be able to work out. So, are these principles that I just showed you on the last slide, are they evolutionarily conserved? Here you see the very famous levels from Pat Churchland and Terry Sinofsky, from the central nervous system up at the top down through successively finer layers till we get down to the individual molecules at the bottom. And we should look at what it is that evolution has conserved. Okay, sweeping up from the bottom. Molecularly, there's a very small number of essential proteins and other molecules that are not, sometimes some of them are specific to neural function, some of them are available for general cell biology, but tend to be used in the same way in nervous systems throughout the animal kingdom. Looking up one layer, there's a very small number of excitatory and inhibitory synapses. There are very, very few neurotransmitters, and there's a small conserved number, conserved in sequence and in structure, of membrane proteins that help carry out synaptic transmissions. Up above, neurons look very, very different from one another in structure, but the number of basic types is very, very small. Enormous quantitative variation in the properties of the neurons, electrical, anatomical, functional, but really they're qualitatively, they're all quite similar, and the neurochemistry is very, very similar. So, but in between, at the network, at the layer of the network or the circuit, are the circuits, really the microcircuits, and the computations and transforms that they mediate, are those similarly evolutionarily conserved? Is there a small number of canonical types? Up at the top, which you can still see, the goals of the nervous system, the componentry, whatever it is that nervous systems do in the life of organisms, that's similarly conserved by evolution. So it's an intriguing question to see if this mid layer is also something that ends up with a small number of canonical properties. And to repeat, we're talking both about the computations or transforms that are performed and also the circuitry that performs them. So there is some complexity, there certainly is some variability, especially to within the networks. But what is it that might be neuromorphically essential? What's common to nervous systems? What's characteristic of nervous systems that might inform our thinking when we start asking these questions? Well, nervous systems have complex connectivity. It's more complex than that of artificial neural nets, which tend to be very, very simple. There are 3 or 4 different ways in which they are connected to one another. But in the brain, in brains, in nervous systems, the connectivity is sparse as well as complex. There obviously are microcircuits that are characteristic of different structures of the brain. The circuitry looks different, but perhaps the functions that are being performed are not so different. We need to study that. There is extensive top-down signaling in almost all nervous systems. In addition to the bottom-up ones we tend to think of from sensation through computation to action. Now, brains have many, many more neurons, like artificial neural networks. Artificial neural networks these days tend to be deep with many, many layers, but the brains tend to have fewer layers than functional artificial neural networks these days. Some of them are like wide rather than deep artificial nets, a subset of the top, and different parts of the brain and different nervous systems tend to be wider or deeper, again, perhaps because they're carrying out the functions in different ways. Most artificial neural networks make very little use of inhibition, but the brain does require inhibition. And what's fascinating is within a structure, there's always a characteristic percentage of neurons that are inhibitory, but this percentage varies widely. It's 2%, say, in something like the cerebellum, up to 50% in some other areas, 20% in the cortex. And the types of inhibitory mechanisms and the effects they perform on neurons vary greatly. Those are again tuned. Biological nets, brains, are very, very stochastic. That is, if you look trial to trial in an artificial neural network, if you feed it the same input, you're very likely to get the same output each time. This is not true of brains at the cellular or synaptic level. And that stochasticity is fascinating and may itself have a computational role. And of course, biological networks make use of plastic synapses. That is, the efficacy of the synapse changes over time. Now, artificial nets have plastic synapses or variable strength connectivity, but that's usually only modified in a training period. And once training is complete, those tend to be fixed and maintained. This is not true of the brain, and that's for the reason that we and our brains have to keep learning at all times. And there is no time when we can expect the brain to be stable and fully wired up with no changes in the future. Now, any system that learns has to have learning rules. Any system that computes has to have algorithms to perform the computations. But again, these are things that surprisingly are imperfectly understood, and we really need to get better analyses of these that in order to carry out these questions. So, how to begin? Well, if there's a conserved property of nervous systems that's useful for computation, then perhaps we can ask what's common to all nervous systems. And here's a list of some of the things. This expands on some of the stuff I said earlier. Nervous systems fire irregularly. The synapses are stochastic and plastic. Essentially, all nervous systems process information in two different ways, through digital spikes or action potentials and through analog or graded postsynaptic potentials. And they all have in between these two a nonlinear transform. And so one question is there a neuromorphic computational advantage to this? There certainly is for artificial neural nets, because artificial neural nets that don't have a nonlinearity built into them at every level, at every layer, don't work very well. One thing that's striking to a cell biologist is that neurons post-development are very, very long-lived. Is this persistence essential? Non-neuronal cell types aren't like that. They turn over perfectly happily. So your red blood cells or your bone or your skin turns over quite frequently without any change, but for some reason, neurons don't. What's the advantage there? Also, neural circuitry leverages very high fan-in and fan-out, the selective balancing of inhibition I mentioned a moment ago. And so this forms a dynamic assembly, but it's stable at any given moment. And that's really quite remarkable that evolution can provide dynamism and constant change while keeping the system stable. Another characteristic of nervous system is that an enormous amount of processing is done by local nets. And the local nets process information, but they're modulated or coordinated by long fibers from other local nets. And I list here some of the things that may be made possible by this, but I want to stress that the connectivity, the long distance connections, come from the bottom up, which seems logical, but they also come from the top down in the nervous system, and they're also lateral. And without this distant connectivity constantly informing the local processing net of how it should be doing its computation, it doesn't work. And again, neurons vary in their connectivity, their excitatory inhibitory balance, how they integrate, and so on. So brains do not have a single kind of processing unit. Okay, so these are some possible candidates for computational essentials. But there are also properties that are common to all neurons that are cell biological. They're important for function, but they're unlikely to be computational essentials. And I list some of the basic cell biology. Without this, the neurons will die, but these are not computational. These are just keeping the system working so it can make use of those special computational properties. And there's also a lot of diversity within and among nervous systems. And I'm going to very briefly show this because this is not an audience of neuroscientists. Different synapses and the neurons that make the synapses are tuned in different ways. The structure of neurons vary and the circuits are tuned. What the synapses do, where on the cells the synapses are formed, all of these things are quite specific. The dendrites of the nervous system are quite specialized. Those are the inputs. And I think these are drawings from Santiago Ramón y Cajal, the great Spanish neuroanatomist, assembled by my friends Kristen Harris and John Fiala to show differences here. And the dendrites are the parts that are shown in the gray arrow and above and a little bit below. And you see on the left, they can be very, very small and selective and take tight inputs from the immediate area. In the middle, this hippocampal pyramidal cell with an extensive dendritic tree is actually sampling the space in which it's located. It's only getting input from about 3 to 5% of the neurons that it could reach with those dendrites. So it's quite selective as to the connections it's making. And on the right, the cerebellar Purkinje cell has this giant sheet that can get hundreds of thousands of inputs. And this one is indiscriminate in the sense that it makes a connection to every fiber that comes anywhere near it. So the way in which neurons compute depends on the circuits in which they are placed. And here are some classic examples. On the left, this is the canonical excitatory cell arrangement in cerebral cortex, showing how pyramidal cells make connections to other pyramidal cells and different parts. In the middle, the cerebral cortex has several kinds of interneurons, and these different interneurons are shown making specific connections on parts of pyramidal cells or of other interneurons. On the right, the circuitry in the cerebellum, and I showed you the Purkinje cell dendrites just a slide or so ago. This combines both excitatory and inhibitory cells. And it's a much simpler circuit, but it's one whose purpose still remains incompletely understood. And there are several different candidates for the computation being performed by this. So one question that we also need to think about if we're going to be looking for canonical connections is what isn't common to all nervous systems. So given this diversity, there are features found in some nervous systems, but not in others. If there are reductionist computational essentials, they're unlikely to be one of the ones listed on this slide. So we do not have all-purpose neurons or one fixed balance for excitation and inhibition, nor is it merely an emergent property of having large numbers of neurons, nor is a neuron a simple atomistic object like in artificial neural networks, because neurons vary widely and some are quite complex single neurons and able to process information. And neuronal firing is highly variable. I told you that to a fixed stimulus, the nervous system never responds exactly the same way, like Heraclitus's river. And here is a stereotyped stimulus being delivered, and these are recordings from 6 electrodes for the 10 different trials here. And you'll notice that for each of the 6 electrodes, the 10 different trials show very, very different results. Now, this variability may be intentional. There may be deterministic input that we don't know about. It may be noise. The very presence of noise may aid calculation. We don't know. Now, here's a fundamental problem that we have. We can record from thousands of neurons in biological neural networks. This is already 10 years old, and that was showing reliable recording of 200 neurons or so. But these multi-electrode techniques and these arrays have gotten even better. And we can now record from thousands and thousands of neurons, but we can't predict the firing of thousands of neurons. If we understood what I said was the problem, how do nervous systems compute and what is the circuitry that does the computation, then we could predict the operation of the nervous system. Now, this is easy for electrical circuits with thousands of components, but it's not true for more than a handful of neurons. And even for those, it's not true for more than a handful of seconds. Also, we can simulate exemplar nervous systems, artificial ones, resembling the brain with very fixed tuned parameter values, but this doesn't work for real specific neurons or circuits. And this is because individual neural and synaptic parameters vary in ways we can't easily measure, and there are too many of them to model exactly. And this raises another question. The brain developmentally selects and tunes parameters other than synaptic strength, but we don't know how. The slow process during development is the brain's first credit assignment problem. Credit assignment being the problem of knowing what to change deep within a network to get a desired result to learn. And then tuning individual synaptic strength during adult life, classical plasticity, is the brain's second credit assignment problem. So how do biological nets perform these two credit assignment problems? Neural networks need to do it to function, to stabilize, and to tune synaptic weights. Any neural network does. Artificial neural networks do it most famously by multi-layer backpropagation, but I already told you that's non-neuromorphic. And many groups have listed proposed alternatives to these. Again, here are the two different properties. Um, I also should mention some clinical relevance. The developmental selection form of credit assignment is disrupted, and the result for that are some very early autodevelopmental disorders. Secondly, there are the individual tuning that is altered in later onset neurodevelopmental disorders that show up, say, between the ages of 17 and 25. As a fascinating footnote at the bottom, there's one transcription factor called MECP2, which manifests early as a gain of function defect for that first problem, but as a loss of function, it affects the second problem here. So the same molecule may be involved in both of these. So here are some of those alternatives I talked about here. You know, the biological neural network local circuits may be pre-programmed for plasticity. There's something called synapse tagging that I won't have time to get to. Is there a local process or a global excitatory inhibitory balance that can do this? Do astrocytes receive global signals and adjust local? Some people have proposed a solution called neuroevolution for this. I don't have the time to go into them, and I'm going to zoom through the slides that present it. Here's a question of a neuron that makes two different connections. The neuron is shown in red between two green neurons here. And in fact, tagging of one output site rather than another can mean that that is functional and will change with a global signal, whereas the other one won't. There is in the possibility of glial cells. The glial cells are shown here in blue. The neurons are shown in this tan color, presynaptic endings on the top, a little bit of the postsynaptic neuron below for each of these four diagrams. And the neurotransmitters, in this case, glutamate, the excitatory, GABA, the inhibitory neurotransmitters, are recycled for further synaptic transmission through the glial cells. And glial cells may make more connections than the neurons and may receive signals to control synaptic strength. And I'm going to skip over this, which shows basically the same sort of thing we saw below. So many groups have proposed various neuromorphic suggestions linking artificial and biological neural nets for credit assignment. Other people have reported properties of BNNs that might be computationally useful or essential. And as these examples scroll majestically by as we go from the upper layers down to the lower layers, this simply shows that there are a large number of potential candidates for an evolutionarily conserved mechanisms for computation and mechanisms for credit assignment. I want to just talk about one very briefly. The first one here, which is shown in red, this is recent work from Chuck Stevens' lab at the Salk, showing that in several different sensory systems, in mammals and invertebrates, maximum entropy coding encodes and decodes the largest number of possible choices that the nervous system has to make given the number of cells used for the task. It's highly efficient. My enthusiasm means that I've run late, so I can't talk about it. But here's a credit assignment one. This expands on what I saw about astrocytes, even including the control of nonlinearities, in addition to the amount of neurotransmitter that's been advanced by a couple of labs. I'm not going to talk about the fly odor coding, but I refer you to recent work from the lab of C.F. Stevens for this. Instead, I want to finish by telling you about the Encubed project here for looking for neuromorphic computation. Now, I pulled these 5 books from my bookshelf. 6 books, sorry. 5 which can do something and the 6th which can't. They all present circuits, but the one on brain microcircuits, Gordon Shepherd and Sten Grillner's, the arrows pointing to it, is not like the others. It's the only one where the anatomy, the function, and the connectivity of many microcircuits are laid out in beautiful detail, but not the computations they perform. Why is this? So why can the electronic circuit books do this? They can take the circuit on the left and predict from that the function as shown on the right, or they can take the pattern of activity shown on the right and reverse engineer the circuit that's doing that, called system identification. So they can do that, but we can't. So are neural circuits identifiable in the same way? And as it says here, electronic ones are. The third bullet point says that there are a few neural circuit operations or calculations. I mentioned 1 or 2 of these earlier. Weighted summation, leaky integration, gated, various patterns of inhibition, lateral inhibition, surround inhibition, non-isomorphic, anisomorphic surround inhibition, which gives you direction sensitivity, response sharpening from surround inhibition, gain control, winner take all, persistence of a signal, delay lines, attractors, even some predictors, nonlinear analog to digital and digital to analog converters. We know neural circuits do this. But if we're thinking in terms of electronics, where are, say, the half adders, or where is something that can demodulate a signal from a carrier? Now, one possibility is we can't detect other than the elementary building blocks and find the serious functional circuits. Well, maybe stochastic firing is a confound, and that makes input-output analyses hard to do. Now, one question is, are neurons just too diverse or too specialized to form canonical circuits? All neurons are variants of only a few basic types, and this is also true of electronic components. So on the left is a listing of some hippocampal inhibitory neurons. There are many different kinds shown there, but they all perform the same basic function. And this is just like the many MOSFETs on the right, not quite interchangeable. You can't plug one in for the other, but all working the same way and all covered by the same symbol. Some people say, I'm advocating consideration of and search for potential evolutionarily conserved mechanisms, but maybe that isn't the case. Maybe all the circuits and computations are just evolutionarily ad hoc, and there is no general circuitry nor canonical, no general theory nor canonical circuitry. Evolution just builds ad hoc, and it's different everywhere. Or computability may just be an emergent property of any circuitry that sums and transforms. So do we care? Some people say they don't care whether it does it or not. I care. And so at the Society for Neuroscience Symposium about a year and a quarter ago, where I introduced this topic, I asked the 100 or so attendees what they thought, and the voting among the neuroscientists was 10-1 in favor of canonical circuits or algorithms. So I want to wrap up with what I want to do about this. Many different labs work on this problem, but what I'm proposing is not something that any single lab can do. To answer a general question requires collegial, parallel efforts by many investigators using different techniques in several preparations. And so what we need to do is to get together. We're going to be planning a multi-day NeuroMatch meeting of this to get this started. And we'll know that this is going to work if at least half the people we invite say, well, I'm already doing that or already solved that problem. And then once we get people aboard this, what we want to be able to do is to encourage funding agencies to support adding explorations of computational capabilities to the ongoing research from many of those labs and those different preparations. And of course, because we're forward thinking, we think all data, code, algorithms, and so on should be open source and freely available. And this would help launch this field and allow other funding agencies to decide. So you're hearing something that's just at the beginning of a new idea. And here are some of the people who contributed to this at symposia in 2019 and in 2020 for this. Of course, the SFN symposium was not just a few months ago, it was a year and a few months ago, because we didn't have one meeting in 2020. But all of these people contributed to this, and I'd like to thank those groups who've collegially helped build this idea along, and now I'm ready for questions.
Dan, thank you so much for that overview of neural networks and what kind of computing they do. And this is just clearly an interesting challenge, which is, it's interesting from the context of data science, which is, so often deals with thinking about how to statistically analyze systems, for example. In this case, you're really trying to take it to the next level, say, perhaps using the knowledge you gain through a data science approach to inform how you might create synthetic models of biological systems. So this is very pleased that you were able to present this to us. One of the things which one of our participants kind of raises a question, I had the same question myself, is that very often these neural networks, the goal, it's claimed is, pattern recognition. We want to be able to recognize patterns. But really, is that what we really want to do all the time? Or really, are we trying to understand the foundational properties of these biological systems? And I think you're a fan of the latter and not the former. And I also want to kind of ask in that same context is, as a result of that, do these artificial versus biological neural networks, do they pass something resembling any kind of a Turing test that we're aware of?
Well, okay, so the first question is, well, people say that these networks are good at pattern recognition or pattern identification because that's how they test them.
Right.
And I think something that I didn't say explicitly, but I probably should, is what you think a system is doing depends upon what question you asked it. But I don't know what it was that evolution designed the system to do. We know what we can do, but we don't know how, but we can do it from introspection. We can do it by observing others, either others of our species or experimental animals, but that doesn't tell us what's actually being done. And the purpose of having a group of different investigators working in different preparations is to say, for someone to say, well, I think that what the microcircuit that I'm doing is computing is the following. And can somebody test out that idea on their preparation and see if that's something similar to it? So that's how we start looking for what might be a small number of canonical computations. So I don't know what it's there for. Now, the other question, you weren't specific about the artificial versus biological and the Turing test. I showed an example. The classic ones are unfortunately ones that are used for pattern recognition and for pattern discrimination. Those are the ones that mimic the visual system. And that is a form of Turing test in that you can give a primate or you can give an artificial neural net the same inputs and allow each one will make the same, mostly the same selections and discriminations. So are you willing to accept that as a Turing test? What we're talking about, of course, is something at a very simple fundamental level. And so I think the Turing test you want is to be able to identify a computation that you think a network of 80 or 100 neurons is performing. And then you should be able to feed it inputs and predict what the output is. That is not quite a Turing test. That is a test of a model. And I think that's as close as you're going to get when we're talking about phenomena at this level.
You know, it's another thing is as you were presenting and talking about some of these kind of foundational building blocks, the rules or the kind of the behaviors of these neurons in these systems, I'm reminded that it's been 50 years since Conway's Game of Life kind of emerged on the scene. And many of us who are of a certain age monkeyed around with those simple rules between neighboring cells in a network to try to produce interesting behaviors out of very simplistic rules. And I'm kind of curious if some of those, I mean, not of course the same rules, but would you hope that a similar sort of, 3-5 rules might be underlying what we see in very complex systems?
It's even better than that, because Conway's Game of Life is a special case and a late special case of cellular automata.
Yeah, exactly.
And the cellular automata that were first looked at in the 1940s and early 1950s by Johnny von Neumann and others were consciously thinking about nervous systems and computing at the same time. And they wanted to do their first look at cellular automata was actually to build a Turing machine. And I think they had one which had, 39 rules, and that could compute absolutely anything. And so they could build a Turing machine out of cellular automata. But again, this was a time when nothing was known about the brain in the terms that we know it now. And the synthesis between cellular automata, computers, and nervous systems doing processing has been with us for what is now almost 70 years. So the links are intriguing. The game of life is just a very, very simple thing. It would be great fun if we discovered that some of neural development, I mean, if you think about this in terms of an electronic analogy, the brain is wired up from a parts kit. These days, electronic devices are all three-dimensional. And you lay down transistors and capacitors and resistors in multiple layers, which is similar to the way neurons go. In the brain, our excitatory inputs the neurons come from the subventricular zone. Our inhibitory ones fly in from the ganglionic eminence just at the right time from a different parts bin. And then they assemble themselves. And that's part of that first credit assignment problem that I talked about here. So there are always going to be engineering questions. It would be fun if some of the principles used for those inhibitory neurons to make some of those connections could be shown to be in some ways isomorphic to some of the rules that cellular automata use. I have no idea if that's true or not, but it shows you how thinking about the brain from this perspective can cause different people. And I hope somebody listening to this is taking notes, saying that would be an interesting problem to work on, how this can inform what work should be done.
One final question for you, Dan. What role does data science play in achieving some of the goals of what you described for us?
It's essential. And I should have made this explicit, because when I talk about people making comparisons from laboratory to laboratory, that requires the easy exchange of data. You can say, this is what my neuron does, my neural network does. And does your neural network do this? And that's not possible without effortlessly taking the algorithm and the data, extending it. And you and I, Jack, worked on this years ago for the Human Brain Project, when we tried doing some of the first fundamental rules for data exchange for the neurosciences. And without that sort of effort, what I propose would not be possible.
Well, Dan, thank you so much for
this is fun.
This is so thought-provoking. I could go all day, but I want to thank you so much for sharing this with us. This was really inspiring, and I hope that our audience was inspired as well. Thank you so much. Thank you, everybody, who joined us, and enjoy your weekend. We'll see you next week. Thanks. So long, all.